Reverend Principal, eminent guests, respected faculty, distinguished delegates, and participants, welcome to second day of International Conference, Role of Life Sciences in Vision 2047. Today we have eminent speaker, Dr. Sougata Ghosh with us from Rajkot University. May I call upon our today's chairperson, Dr. Ritesh Khandel, Head of Microbiology Department, to welcome and honor him. I request Dr. Ritesh Khandel to welcome him. Welcome, Dr. Sougata Ghosh. Thank you, sir. Now I request Dr. Ritesh Khandel to introduce our speaker. Thank you.
Namaste. On the behalf of the Science, we welcome Dr. Sougata Ghosh. He is currently working as an assistant professor in the Department of Microbiology, School of Science, at the University of Rajkot. He is also visiting professor in the Department of Physics, Faculty of Science, SS, the University Bangkok. He is active researcher in nanomedicine, having obtained his bachelor's, master's, and PhD degree in microbiology from the University of Pune. Now, this university is known as Savitri Pune University. He is also a postdoctoral fellow at the Northwestern University, USA. Sir has filed 8 patents. He published 271 publications, including 10 books, 168 book chapters, and 92 international research in various languages. Sir has also is a very proud moment. Uh, 6,500 citations, H index of 43, and IQ index is 107. He is listed among the world top 2% scientists from 2005, 2025, 2024, 2023, and 2020 as a released by Stanford University, USA, and NASA. Sir is also known as a research person and invited speaker in 160 international and international conferences, symposiums, and workshops. The most notable being ICMR 2023. Dr. Ghosh is the assistant editor of the Portals Chemistry Directory. It is current microbiology experience. He is also a member of the Molecular Boards, reviewers, team of the 127 Indian countries. Sir was principal investigator of the DBE Government of India. He has also received several prestigious awards like Junior Research Fellow in 2011 by Institute of Bioinformatics and Biotechnology, University of Pune. Sir is also awarded by Junior Research Fellow in 2012. Sir is awarded by the Bharat Gaurav Puruskar in 2020. You welcome, sir. Now I request sir to start his session as a He will speak on novel level medicine from the Econo Medical Plan for the Sustainable Healthcare. Good morning, all. Thank you, Dr. Pandey, for the kind introduction. So, as you can see, today's talk, the topic of today's talk is novel nanonutrition from ethno-medicinal plants for sustainable health. So, we have botany over here, we have plants, then we have nanotechnology, that is physics, then the synthesis, stabilization, characterization, chemistry, and application in the microbiology and biotechnology. So you can see that the research has now become more interdisciplinary. So we will be talking about interdisciplinary research. So coming to the history of nanotechnology, as you can see, father of nanotechnology is Sir Richard Feynman. So Richard Feynman wants to organize plenty of new metabolism, and it has an implication to enter the new field of physics. So you can see over here that there are different ways of nanoparticles in this. So if you see the nanoparticles, those are very, very small particles which are in the range of 1 to 100. Now, there are two ways for synthesis of these nanoparticles. One is you have a very big bulk metal and so we have a big metal over here, then we break it down into smaller and then further break it down into very small particles from 1 to 100 micrometers. So this process from the bigger to smaller, this is called top-down approach. At the same time, there is another process. In another process, we have very big small particles, we can see on this right side over here, and these particles they grew up into 1 to 100 nanometer. So we call it this process is called a bottom up. Now the question is, do we take these nanoparticles onto the laboratory? Whether we can only synthesize these nanoparticles or we can get elsewhere also. Makes sense. So here you can see there is a butterfly. It is a male butterfly called Dixonia disco. It is a male butterfly. As you look carefully, you can see four different colors in the. So the first color is the black. Next, you can see the blue color. Then you can see light brown and dark brown color. So if you look into the light brown and dark brown color of the butterfly, now see, we have to resolve the thing. So it is now research is completely multidisciplinary. So you can only take your subject, your stream with the content of the student. So you can see the brown color spots that are going same pair and same patch. So what are these brown color spots? It is a way butterfly. It releases a chemicals which are called pheromones. And these pheromones are used to attract the female. Now, apparently, we may think that these wings they have colors, but actually all black, blue, light brown, or dark brown, they do not have any colors. Now, if you look into this dark brown color, you have columnar cells with hollow interior. On the other hand, if you see over here the light brown, it has fibrous hair-like structure. On the other hand, if you see this black, they are parallel rows. And on the other hand, if you see a pool, those are cuboid, all the cuboid. So all these structures they work like nanoprisms. When the light passes through these nanoprisms, then there is diffraction and you get these colors. So actually, the wings of the butterfly they are not colored, they are colorless. But because of this, because of the diffraction, you get the color. So that means these nano patterns are very important and we get them in nature. So what we do in our lab, we try to produce the nanoparticles from nature. So what are its applications? Now those nanoparticles which we can derive from nature, they are applied in different fields like electronics, semiconductors, catalysts, biosensors, and even solar cells, cosmetic paints, and drug delivery systems. So let us come to the next slide. This is the slide where we are showing different types of nanoparticles that we have synthesized in our lab. These are presented and published. So first you can see the gold nanoparticles. These are the gold nanoparticles first. Second are silver nanoparticles in the hexagonal. Then it is like our coin. Gold is in the center and silver is in the outside. So it's the gold core silver shell nanoparticle. Finally, these are all from plants. All from plants. All these nanoparticles. Next, we have copper nanoparticle over here. Here, we have magnetic nanoparticles. Magnetic nanoparticles, which are not integrated. These are gold nanoparticles. These are gold nanoparticles, liposomes, palladium nanoparticles, and zinc oxide nanoparticles. So different types of nanoparticles which are synthesized from nature and used for medicine. And these are all related and published. Next slide, please. So let us come to nanomedicine. What we are doing in our lab and how these particles are used, how they can correlate your research or you can develop your own research program. Next. So this is a plant. Whether we have seen in general also in Himalaya region, in different hills. This is like wheat. This plant is called Telescoria cultivar. Probably you have seen this in your backyard, but you have never noticed it properly. If you planted it might be a backyard of your house. This plant is called Bharati in Ayurveda. In Ayurveda, it's called Bharati and this is Diaspora. So you can see this air potato, in English it's called air potato or yam. One single air potato has so many and therapeutic activities. So they have any disease, maybe cancer, ulcer, inflammation, any kind of disease, infections, even snake bite, anti-tumor, antifungal, each and every kind of disease this plant can use. So it's such a powerful medicinal plant called Diaspora. Next. So, what is the life cycle of the Diaspora culture? Now, if you are from what age, you will be knowing that most of the plants which are monocotyledons have a parallel lineage. This is very unusual plant, which is monocotyledon but has reticular lineage. It is an exception. Diaspora. So generally, the thing is like this. It looks like the heart shape, the now, this is generally the month of March, April, it starts growing, it starts sprouting. Imagine the growth rate in the month of May, it starts growing 15 centimeters per day. एक दिन में 15 centimeters it grows. And if you have a support system, if you have some electric pole or something, it will cover up the entire thing within 1 month. Everything will be covered. So it is called an irrigation by Florida in US. It is such a powerful growing plant. In the month of July, you get these kind of vegetative organs, which are called the tubers of the diaspora cultivar. And these tubers are generally used for the propagation. The plant never produces seeds or fruits, but these tubers are the vegetative propagating organ of the plant. In the month of September, there is flower, male and female flower, but they are sterile. These flowers do not pollinate, they do not form a fruit, they do not form a seed. But in the month of September, we have this flower. In the month of November, there is a dieback mechanism. There is a even if you give water, if you give fertilizer, you get the any number of micronutrients, macronutrients, the plant will never survive. Let's say November after, the plant will die. It will die completely. What remains on the ground is those tubers. Those tubers remain viable for 1 year. So for 1 year, it is viable. Generally, I visit the tribal belt of different states. Although I am from the from West Bengal, but I stayed 15 years in Maharashtra and I was working from 2018 in Gujarat. So whenever I get a chance, I try to visit different types of tribal belt and I start researching and start how these plants get used. So I collected this plant from a region called Jauha in Nasik region of Maharashtra. So if the people those tribal that not affected in the Nasik region, in Jauha region, they do not get regular rainfall. So वहाँ पे agriculture is practically unavailable. So it is agriculture unavailable land. When they eat this diaspora instead of one, they do not feel hungry for 2 days. Those things are on the book. Because it has slow and sustained release of starch. They do not have any kind of diseases like high pressure, low pressure, diabetes. They do not have these kind of diseases because they come consequently consume this food. So now, when we collected this plant, I kept it in a cover or you will put there for 4 5 months I just cover. In the way, when we open my cover, I found the plants that started sprouting up, they everything was. The plant is so robust that it does not require soil, it does not require water, it does not require anything. On the other hand, I have the newspaper. Newspaper का cellular news करके the plant started growing. So it is as robust as Varama. So that's why it is called Varanikan in our Ayurveda. Next slide, please. So, why we are using this plant for our research? There are different other methods of intelligence. We have physical method and chemical. But whenever we are using physical method, we have to use certain high or very abrupt conditions like high toxic or high heat and high pressure, all these things we have to use. In case of chemical systems also, we are using certain stabilizing agents, certain reducing agents that are toxic. So in that case, we are synthesizing the nanoparticles for electric applications, solar cells, photovoltaic cells, so but if we are using for medicine, then it renders bioincompatible. The nanoparticles are toxic. If the nanoparticles are toxic, then we cannot use them for the therapeutic purposes. So that's why we shifted to green synthesis of the nanoparticles. Next slide, please. Okay. So, why we are using diastolium? What is the reason that we are using diastolium? This plant, diastolium, it has phenolic, it has flavonoids. So both these act as reducing agent. It has ascorbic acid, it has citric acid. They act as reducing agent as well as stabilizing agent. It has saponins or diastolium that act as stabilizing agent. We do not have to add anything from outside. We just mix the diastolium extract and when the particles are synthesized, they are stabilized, they are stabilized in 1 year. That is the stability of this diastolium. Now, moreover, since we are not using any physical or chemical agent, more biocompatible and the activity is enhanced because if the diastole is able to synthesize metal 1 and metal 2 and combine them together to form a bimetallic or fragmental. So that was our aim for using diastole. Next. So what we are doing in our lab? We have a plant diastole. Now we will connect it to another plant in successive slide. That is the media blocker. That is also very interesting plant. Next plant is Lambago say that it is that is also very important Lambago seneca. So what we do generally, we take roots, stem, leaves, flowers of different plants, and then we prepare the extract. From the extract, we synthesize different nanoparticles, then we prepare conjugates, we do characterization. At the same time, we extract and then do sequential extraction of the extract with different solvents, and then we try to purify those compounds with separate type of PLC, HPLC, follow HPLC. Then we do the characterization with the help of UP, NMR, FTIR, and different methods. And then with those chemical compounds, we try to optimize the temperature, time, concentration, and develop optimized. So this is how we find the patent of optimized green synthesis of food. Now, when these nanoparticles are produced, then we do different types of biological activities like antimicrobial, antibiotic, anti-cancer, antifungal, different other activities, and then we develop the nanomedicine. Next one. So let us come to the first stage where we prepare gold nanoparticles. Now, as for here, if any of you consider if you take up rather chance, please see the ingredient. So now it is called previously it was rather chance, now it is called rather smart and why it is rather smart and because this diaspora valvula is a component of the carbon chemicals, and this is used previously for synthesis of swan of plasma and detergents. So this gave us an idea that today this Varanika can produce or may produce gold energy. So what we did, you can see over here, we have taken this bulb, cut it into pieces, dry it, then you make a powder, and then you extract. Before you start any synthesis, it is very important to know what is present, what is the phytochemistry of this extract. Because if you take the diaspora in Gujarat, if you collect the diaspora from Maharashtra, if you collect it from Nepal, if you collect it from Florida, you need phytochemistry in there. Every time the phytochemistry varies, the nanoparticle is vary. The structural properties, the functional properties will vary for the nano. So it is very important when you synthesize, when you start the synthesis, you should know what is the phytochemistry of it. So in the beginning of it, we take a biological acid and know the total thermal content of the extract. Then we found starch content of the extract, total reduced sugar, balanced jelly, ascorbic acid, and citric acid. And to our surface, we found each of those components are present in the diaspora. Why only this component? Because these components are very, very essential for not only the reduction of the metal ions to metal nanoparticles, but the stabilization and the energy. So that is why we started investigating this. Once we confirm these are present, then we started mixing this extract with gold chloride, and within 90 minutes, within 1.5 hours, that gold nanoparticle synthesis was complete. This was one of the fastest synthesis. Within 1.5 hours, gold nanoparticle synthesis was complete. That is 90 minutes. How we confirmed it? With the help of UV visible spectroscopy, we had a peak at 540 nanometers. So this peak was at 540 nanometers. So there was a big period, and after 90 minutes, that came to threshold, after that level to increase. So after 90 minutes, the gold nanoparticle synthesis was complete. Next question is optimization. What is the rate? What are the important parameters that are responsible for the nanoparticle synthesis? So we optimize the gold ion, the gold nickel salt concentration. Now here you can see this is fluorouric acid and nickel concentration. The concentration varies from 0.3 to 5 millimolar. Now for sulfide you saw we are using 0.3, the gold nanoparticles are gray in color. As we increase the concentration, the color intensity will be increasing, but it's not yet. Not only the intensity, but the entire color change. As we were changing the concentration, the color of the nanoparticle changed from gray to this very light red, then ruby red, dark red, orange, and then golden yellow. That is each concentration leads to the change of the size of the nanoparticle. As the nanoparticle grows in size, it makes it metallic golden yellow color. If the nanoparticles are very, very small, they are gray in color. If they are around 100 nanometers, 50 to 100 nanometers, they are ruby red in color. So as you change the concentration, not only the rate of synthesis change, but the property, the structural properties of the nanoparticle started changing. So we found that 1 millimolar builds over here. 1 millimolar. At the same time, we started changing the temperature. We raised the temperature from 4 degrees Celsius to 50 degrees, and maximum synthesis was at 50 degrees Celsius. So, next question was, what is the gold conversion or what is causing something different in something else? So we confirmed with therefore FESS, that is emission electron microscope. We found very, very small gold streaks, critical gold nanoparticles. They were coming to form small triangles. Smaller triangles were coming to form bigger triangles. So there was a hierarchy from sphere to triangles. Also, we found gold nanohexagons. Now, in order to confirm size and shape is okay, but what is really gold, we need another characterization that is called EDX, energy dispersive spectroscopy. So when you aim to have a good international paper, a good publication from your research, you have to include all these kinds of analysis to confirm whether it is gold or not, or any kind of result. So in EDS spectra, we confirm international gold. After that, we wanted to measure the size of the nanoparticle. We call hydrodynamic size. It was measured using DLS, dynamic light scattering, and you can see we have two types of size. So first side is this 11 to 30 nanometer, as I told you, very, very small spherical nanoparticles, so those are 11 to 30 nanometer, and also 50 to 300 is bigger triangles, bigger hexagons, where 50 to 300 nanometers. So you can see, so whatever you observe in the HCM, it was reflected in the DLS. So this confirms what kind of nanoparticles you are. Then we found that we confirmed the crystal meeting with the help of XRD. This is the XRD. After this, we wanted to confirm which functional groups are responsible for the synthesis. So we did FTIR. So in this FTIR, when we did FTIR, we found the orange group, the hydroxyl group, so the phenolics or the flavonoids. They are the most important group in the synthesis. In the beginning, you can see that line is the hydroxyl group side in the beginning. DBT, direct nuclear algebra dual extract in the beginning. In the middle of the synthesis, you can see this red line, and after the completion of the synthesis, you can see the blue line. So slowly, this speed is reduced. This speed is so reduced because the four groups are getting involved, getting consumed during the synthesis. Now we confirm that it is synthesizing gold. Next we thought, since it is synthesizing gold, is there any possibility that it will also synthesize silver? So that's why we started synthesizing silver nanoparticle using silver nitrate. So in the exit, you can see there are two plants. So silver nitrate was completely blank in color and transparent. And after that, when the synthesis was completed after 5 hours, you can see a brown color was there. So even the silver nanoparticle is UV visible spectrum and will confirm that the silver nanoparticles are there. We optimize the parameter, we optimize the silver nitrate concentration, which shows 0.7 millimolar of base. We optimize the temperature also, which shows 50 degrees of base for silver. Now, what was the size of the silver nanoparticle? We did a microscopy, which is called HRTPS, high resolution transmission electron microscopy, because it is so small, it was not visible with the electron microscopy. So we did HRTPS, high resolution transmission electron microscopy. And you can see these are very, very small nanoparticles spheres, which are around 8 to 20 nanometers, and these are hexagons, which are around 50 to 70 nanometers. We confirm with the BDF and DLS also confirm the size. 30 to 20 nanometers. Smaller particles are also present. As well as 75 nanometer bigger particles were also present. We confirm the crystallization of this silver nanoparticle with the help of excited and also confirm the reaction with the help of ATM. Now, we wanted to develop the basics. This was a synthesis part and calculation part. So, when you go for your research paper, your research activity, and also in the beginning, what you have to first finish up your synthesis and calculation. Now, we are coming to so here we started with all those who are from field of microbiology or biotechnology, probably you will understand this. This is very kind of MIC examination. You have different concentration of the drug, the agent, and then you see whether it is able to inhibit or not. So we check this activity against 14 different bacteria. One for 14 different bacteria. Different concentration of the drug. And you can see this is 1000 microgram and this is 10 microgram. Even at 20 microgram, it was able to inhibit bacteria like E. coli, it was able to inhibit bacteria like Klebsiella pneumoniae. Even at 10 microgram, it was able to inhibit infection and disorder. So it is so powerful that only 20 microgram, 10 microgram, these were because now you know the most important is facing that is MDR, multi-drug resistance. So that was our main aim, whether these particles are able to inhibit these bacteria at a lower concentration, very, very low concentration. So these are the 14 bacteria. So you can see these are the bacteria. Now with this, next slide, with this we created the biggest table of the world in 2012. Now this table has got more than 700 citations. 700 researchers were conducted using this table. So this table will be published in 2002. So what does this table tell about? Look into it, try to understand carefully. Here we have 14 bacteria. Okay, this table has two parts. This is one slide, another slide is also there. There are 14 bacteria you can see at the top. Okay, so you have the bacteria. There are 14 bacteria. Here you have 23 different antibiotics. In this table, you have 23 different antibiotics and 14 bacteria. Now you have three columns for each bacteria. Column A, column B, and column C. What is column A? Column A states the zone diameter or activity of only antibiotic. Sit antibiotic. We have added nothing in this. That is column A. What is column B? Antibiotic plus our silver nanoparticle. How much is the increase in the zone diameter? And column C is fold increase. How many fold the activity of the antibiotic is increasing? Now you can see when we are combining piperacillin with silver nanoparticle, its activity is increasing 3.6 fold for Acinetobacter only. When we are combining streptomycin with nanoparticle, silver nanoparticle, its activity is increased up to 11 fold against E. coli. So people started referring this table all over the world. Researchers started referring this table to form nanoconjugates. Which nanoparticle, which antibiotic, and which bacteria it is showing best. So they started their research. They now don't have to screen because our table is already available. So this was how we conducted this synergy. This is called antimicrobial synergy. Next slide, please. So this is the continuation of the table. So it was one of the very big tables that we published in 2012, and people started referring this table. Next. So then we thought, since this diaspora bulbifera is synthesizing gold, since it is synthesizing silver, what will happen if we combine both gold and silver together? That was our question that we were having at that time in our mind. So then, when we combine gold and silver together, you can see there is a blue line, and the blue spectra was for gold and at the same time, there is a green spectra. The green spectra was for silver nanoparticle. But when we combined together, there was the red spectra that neither merged with the blue spectra nor it merged with the red green spectrum. So this told us, this indicated that something different was getting formed. It is not like that when we are adding silver and gold, थोड़ा सा silver nanoparticle हो रहा है और थोड़ा सा gold nanoparticle हो रहा है. Then we will have got two spectra. Or only gold nanoparticle over it, then we would have got a spectra that merged with the blue. But we were getting a red spectra that was not merging with any of only gold or only silver. So this gave us an idea that something different is getting formed. But at this stage, we did not know kya ho raha. At this stage, we had no idea what was happening. Then we wanted to see the UV visible spectra, and we found that the spectra, the synthesis of the nanoparticle was getting completed after 5 hours. After 5 hours, the peak it stopped. So the peak was formed. Next slide. So here we wanted to now know what was that something that is getting formed. कुछ तो form हो रहा था. That was clear. But ये क्या हो रहा था? What was this? We are not clear at this moment. So here you can see that the nanoparticle had something in the center and something outside. So there was a core and there was a shell. At this moment, we know only that. There was a core in the nanoparticle and there was a shell. You can see it's like our 10 rupees coin. Okay? But what was in the core, what was in the shell, to know that we did another analysis that is called elemental mapping. This is HRTM. Okay? So this is HRTM where we can see the core and shell. But to know what we did another analysis that is called elemental mapping. In elemental mapping, we can give color code to each and every element. So we gave a color code red to gold, and we gave a color code green to silver. So what we found that red color was masked. Red color could not be seen. What we could see was only the green color. Green color was coded to silver, and red color was coded to gold. But gold was masked. So that means gold was inside and green was outside. The silver was outside. So this confirmed that it was a gold core, silver shell nano. So this is how step by step we try to find out. You can see this red core over here. Okay, so this red core is for gold. Next, we confirm whether it is gold or silver shell with the ETS. So ETS gave indeed there was elemental gold and elemental silver. We wanted to know what was the size of the core and what was the size of the shell. So here we found the size of the core was 9 nanometer and size of the shell was 15 nanometer. Very, very small particle, very small. Smaller the particle, bigger the activity. Smaller the particle, more active they are. So that's why it was very, very unique that these nanoparticles were just 15 nanometer in size, very small. If you hold, if you take the particle on your hand, you have seen after 30 seconds, it goes inside your skin like mehendi, and you cannot take it out from your skin. It penetrates inside. You just put a drop on your hand, and within 30 seconds, it goes inside the skin. Next, please. So now, if you see any disease, you consider any disease, may it be cystic fibrosis or periodontal disease or capital bone disease. Remember, a single bacteria can never cause a disease. Any kind of disease is caused by a consortium of the bacteria or a group of bacteria. They generally form biofilm. Next.
So that's why we wanted to see whether these nanoparticles are able to inhibit the biofilm. Killing a bacteria, inhibiting a bacteria is good. Whatever you are doing in lab, that is good. But in general condition, normal condition, it is not a single bacteria that is causing disease.
Condition, it is a group of bacteria that is causing disease, mostly because of biofilm. Why biofilm formation? Because biofilm is highly resistant to drugs. Normal bacteria, planktonic cell, they are sensitive to drugs. But once the bacteria try to gain resistance, they start forming biofilm. And why we have selected this bacteria, Acinetobacter baumannii, Pseudomonas erythrinalis, and all these things? There is a center called National Burn Center in Airoli, Mumbai. If you go to Mumbai, when you go to Mumbai from collecting this bacteria, you can see there is a National Burn Center. Patients are lying down completely green in color. Their whole body has become green of the patient. Why? Because when there is third degree burn, when there is 90% burn wound, 80% burn wound, they practically do not have skin on their body. So there is an opportunistic pathogen that is Pseudomonas aeruginosa. Pseudomonas aeruginosa completely cover up the body and it produces pigment, And because of the pigmentation, whole patient's body has become green in color. And the patient do not succumb to death because of the burn wound, but because of the opportunistic pathogen. So that is why we selected Acinetobacter baumannii Pseudomonas aeruginosa for the antibiotic activity. Now, you can see when we added this gold core silver shell nanoparticle, it could inhibit Acinetobacter baumannii up to 83%. High amount of antibiotic activity is saw for the Acinetobacter baumannii. Similarly, for Pseudomonas aeruginosa, 18% biotic inhibition, E. coli 22%, and case was 50%. So it is one nanoparticle that can inhibit all different types of biofilm causing infections. The next one. Now we wanted to see exactly what is happening. In a test tube, that is a different thing. But exactly in real time, what is happening we wanted to study. So we did the FESCN, that is the field emission scanning electron microscopy. And you can see this is an intact Acinetobacter baumannii biofilm. Intact biofilm. Sorry, the whole surface, the entire surface is covered with Acinetobacter cells. You can see over here, A1. As soon as we added our nanoparticle, within 24 hours, you can see only 6 cells are left on the substrate. You can count only 6 cells are left. Entire biofilm was eradicated within 24 hours. It was so powerful. You can see only 6 Acinetobacter cells are left over here. Similarly, if you see this B1, this is Pseudomonas aeruginosa biofilm. Whole surface is covered with Pseudomonas aeruginosa. When we added our nanoparticle within 24 hours, you can see the whole biofilm, entire biofilm is lost. Okay, it is removed. At the same time, if you look carefully, very carefully, you see, you can see there is a whole formation, there is a pore formation of the bacteria. Bacteria at the surface, the whole body. And because of that whole formation, whatever was inside, okay, whatever cell components were inside, they were coming out. There was leakage of the cellular material, cellular metabolites, and because of that, there was a cell death. So it was so powerful that it was busting the cell wall and it was taking out every component of the cell. There was a leakage of the cellular component, the cells were dying. Next, the C. Next, if you see that is C1. C1 is E. coli. You can see entire surface is covered with E. coli, and over there you can see only one cell of E. coli after treatment, just one cell. You can use the power of this 15 nanometer nanodrug, the nanomedicine. It is so powerful. After this, you can see this bunch of grapes-like bacteria. So you know this is Staphylococcus aureus. So Staphylococcus aureus, even this biopsy after treatment, it was removed. If the cell wall was chopped off, there is denaturation of the cell. We wanted to understand the 3D imaging, what is happening in 3D view. Next slide. So this is another microscopy called AFM, atomic force microscopy. So in this atomic force microscopy, we know two things. In order to know two things, we do this atomic force microscopy. First is the 3D imaging, what is happening, and second is Young's modulus, or what is happening to its elasticity. So you can see over here, A1 is Acinetobacter baumannii biofilm. After treatment, we can see that white one, that is one cell left on the substrate, just one cell left. So it was indeed matching with the observation of the ACM. Here you can see the AFM image of untreated biofilm for Pseudomonas, and this is the treated biofilm of Pseudomonas. The surface is almost clear, only few patches of biofilms are left. Only few patches are left. Here you can see E. coli. All these undulations, all these uprising, these are from the E. coli biofilm. Over here you can see just two cells of E. coli are left. And you can see small dots. Those dots are two nanoparticles that are lying on the surface. And this was Staphylococcus. spherical bricks-like structure. D1, staphylococcus biofilm, and you have biofilm condition. Next one. Now here you can see over here there is the Leishmania donovan. Leishmania donovan. If you have read this, Leishmania donovan causes Kala Azar or Leishmaniasis. It is a protozoal parasite. So this Leishmania, which is causing the disease, this is like this spindle shape, and you have a flagella. This is the intact, undisturbed Leishmania. It is the native Leishmania, the intact Leishmania, untreated. Now, when we added our nanoparticle to Leishmania, let it react for 24 hours, just 24 hours, we let it react. Whole cytoskeleton has collapsed. पूरा जो cytoskeleton है that is collapsed and leishmania is unable to maintain its shape. The leishmania is dying, it is crippled into a spherical mass and finally the leishmania dies. We confirm with the help of AFM also, you can see an intact leishmania untreated. As soon as we added our nanoparticle, you can see there is complete deformation of the leishmanial shell. And then we confirm with the help of DNA fragmentation, this is intact untreated DNA. Over here, you cannot see the DNA, there is tearing. So the leishmanial DNA is getting damaged. Next one. So this was about the infections. This was about the infection, pathogenic infections of nanomedicine. Now, we have synthesized some nanomedicine also for cancer. Now, if you see cancer, a lot of researches are there till date. But actually speaking, cancer has no answer till date. There is no some medicine or vaccine that can actually eradicate cancer or 100% be sure that it can remove cancer. So you can see there are so many different types of cancer. Blood cancer, breast cancer, brain cancer, you name any part, any organ related to cancer for them. So what are the medicines that are available in the market? The medicines like cisplatin, carboplatin, oxaliplatin, then pyriplatin, these are there. But these medicines, these chemotherapeutic drugs, they do not know which one is your body's healthy cell and which one is cancer cell. So as soon as you take chemotherapy, it will go and affect your healthy cell also. So that's why there is multi-organ failure and many patients they do not die of cancer, but because of the side effects of that chemotherapy drug. The next one. So that is why we thought, can we develop a medicine that can selectively affect the cancer cells and not affect the healthy cells. So what we have done over here, we have synthesized a magnetic code. This is a magnetic code, FFCO. This is a magnetic nanoparticle, iron oxide nanoparticle. On this magnetic iron oxide nanoparticle, we added a layer of citric acid. Second one, citric acid. On this citric acid, we added one saponin from diaspora, from the same plant, Varietum, from diaspora. There is a saponin which is called diogenin. You can see diogenin. So we have added diogenin on this iron oxide nanoparticle. But what is the linker? Citric acid is the linker. So citric acid is between the diogenin and the magnetic nanoparticle. Once we synthesize, you can see the size. It is 12 nanometer and this is 21 nanometer. So 12 to 21 nanometer, not major difference. 12 to 21. We confirm with the help of SEM, EM that these nanoparticles are very small and they are aggregated. The next one. So we saw the particle size of this and we found when it is aggregated, only iron oxide is 60 nanometer and iron oxide with diogenin conjugated nanoparticle is around 150 nanometer. We confirm with the help of those who are from chemistry, you might be knowing, you generally know that the functional groups. So we have to confirm whether diogenin was functionalized on this drug. How do we confirm? If the functional groups that are present in diogenin are also present in the conjugated drug, then you can confirm that diogenin is present. And we found that the signature functional groups of diogenin that is in the red spectra, they are matching with the blue spectra. So that means diogenin was conjugated. The next we wanted to see the anti-cancer activity. So there is a cell line called MCF7, that is a breast cancer cell. So what we did over here, we checked the effect of these drugs with nanomedicine on the MCF7 cell. So you can see this is iron oxide nanoparticle, which is bare iron oxide nanoparticle. Nothing is functionless, कुछ भी नहीं है इसमें. Only pure iron oxide nanoparticle. It is having an activity, an anti-cancer activity up to 28%. When it conjugated, when it is a function of the linker, what is the linker? Citric acid. Then its activity increased up to 35%, a slight increase. And now, on this, when we functionalize the drug, the diazoxide, the natural saponin, you can see its activity, anti-cancer activity, it got shot up to 55%, almost 55%. But on the linker, just citric acid, just diazoxide, they were lesser as compared to this functionalized. So we need an assay which is called wound migration inhibition assay. Now you know cancer has different stages. Initially, a benign tumor is formed. What is benign tumor? The tumor is localized at a spot and this tumor never spread. So इसको benign tumor बोलते हैं. But when it progresses, when it goes to the next level, then it undergoes metastasis, it gets converted to malignancy. What is malignancy? This benign tumor, which was able to stay in one place at one time, it has now started spreading. It has started migrating, and it starts migrating to different organs. And you get multi-organ related cancer in different organs. So here our main aim was previous experiment told us that it is inhibiting cancer cell, but only inhibition is not the solution. We have to stop spreading the cancer cell also. So this experiment is called wound migration assay. So I'll explain you this assay. You can see something over here in both the sides. Okay, so this is a lawn of MCF7 cells, cancer cells. Lawn. पूरा MCF7 से भरा हुआ एक lawn है. It's a lawn of MCF7 cells. Then we created a wound. Okay, so there is no cancer cell over here. This is a wound. We call it wound. Not this one. No cancer cell over here. After 24 hours, if we do not treat, if we do not add any chemicals, any nanoparticles, nothing, कुछ भी add नहीं किया. After 24 hours, we saw the lawn is or the wound is closed. There is closure of the wound. Why there is closure of the wound? Because the cells are migrated from both sides. Left side cells are there, right side cells are there. The cells are migrated from the left, cells are migrated from the right, and the wound is closing. Because the cells can migrate. But when we added our iron oxide nanoparticle, you can see there is also migration, but on this region there is less migration, here is more migration. So there is an unequal distribution of the migration pattern. When we added citric acid conjugated iron oxide nanoparticle, you can see the migration is inhibited. इससे migration कम, but still there is migration. But when we added this dioxin conjugated iron oxide nanoparticle, there was least migration. Migration was very low. From the left and right side, the cells were unable to move towards the wound. So the wound was, although it was reduced, but it was not reduced like the control group, you can see. There is a wound over here. Similarly, for citric acid and dioxin also, there was little bit of wound migration inhibition. So how much was the wound migration? You can see dioxin conjugated iron oxide had the least migration. Only 40% migration was there. So around 60% inhibition of the wound migration was there. With others, you have how much migration in iron is? Only iron is 89% migration. So cells are migrating very largely. They are migrating more. In citric acid conjugated iron you have migration up to 50%. Okay, so this confirms that it could inhibit the wound migration. Next. Next, we wanted to see two things are over. First one is it is inhibiting the cancer cells, it is inhibiting the spread of the cancer. Now, if it is killing the cancer cell, What is the mechanism? We know cell death can be either because of apoptosis or because of necrosis. There are two methods of cell death. So we confirm whether it is apoptosis or necrosis. This is confocal microscopy. So in confocal microscopy, we did staining. There are three stains used over here. You can see there is some blue color. So that blue color is Jackie. That is says where the nucleus is there. If there is a nucleus, the nucleic acid will be stained by that blue in color. After that, you have another color that is green. Okay, so this green color is because of this stain. If you can read from that side, Alexa floor 488. Okay, so whenever there is apoptosis, if there is apoptosis, there is a molecule called phosphatidylserine. Phosphatidylserine will come out and it will align in the outer surface of the cell. सिर्फ apoptosis में ये हो. Only during apoptosis this happens. Phosphatidylserine externalization of phosphatidylserine. जैसे ही phosphatidylserine बाहर आ जाएगा, as soon as phosphatidylserine comes out, this Alexa flow can stain it and it forms green in color. And if there is apoptosis or necrosis, if there is membrane damage, then this PI can go inside the PDM iodide and it stains red in color. So red color will be in both apoptosis or necrosis because both necrosis and apoptosis will have the cell membrane damage and PI will go, it will stain it red. But green color will only be in case of apoptosis. So here we can see with IONT chain, you can see both red color and green color, that means it is undergoing apoptosis. So first there is externalization of the phosphatidylserine, it is forming green in color, and then PI is going inside and it is turning red in color. So this confirm apoptosis. So this was confirmation of the anti-cancer activity. Now, one of the most important challenge over here is how can you control the shape of the needle? So as I told you, during vacation, whenever I get holidays, Saturday, Sunday, जब भी मुझको मिलता है, I generally visit a tribal. I prefer visiting tribal. And luckily in Maharashtra, Gujarat, there are many tribal belts nearby, so you can visit these places. So anybody knows this place? Have you seen this hill? This is called Bhimashankar. We have 12 Jyotirling throughout India. This is Bhimashankar Jyotirling of Maharashtra. So if you get a chance, you generally visit Lonavala, Mathera, but when you get a chance, you visit Bhimashankar if you get a chance. In Bhimashankar, you have this plant. You can see this plant. So this plant is here. So when I went to Bhimashankar, like this during this season, I found that cows, buffaloes, goats, literally cattle, when they are grazing, they are maintaining 20 feet distance from a plant. कोई एक particular plant से 20 feet distance maintain करके जो है they are not going near that plant. Cows, buffaloes, sheep, जो भी animals है, they are not going. Then I asked those people, they call them vaidus, जो गांव पे जो treatment करते हैं, those doctors, the village doctors, they are called vaidus. So I asked those vaidus, why these cows, buffaloes, they are not going to this? They told, sir, if you smell the flower today, tomorrow all of your teeth will fall down. आज अगर आप इसके flowers सूंघते हैं रात को, कल सुबह तक आपके सारे दांत गिर जाएंगे. It is so powerful. And this plant is called Dakpadi. So the local name of this plant is called Dakpadi. The scientific name is called media blocker. What is this plant? In Bhimashankar. And this plant grows only at a height of 3,394. If you start bringing this plant to 2,500 feet, it will not grow. It is an altitude indicator. 3,000 feet के ऊपर ही ये plant grow करेगी. You cannot grow it in the foothills. आप घर पे जाके balcony में इस plant को grow नहीं कर सकते. It is impossible. So then I asked the village people that those values and village people. तो ठीक है, ये तो बात गिर जाता है. I can understand. But is there any other benefit of this plant? They don't, sir, every Monday we have a village market.
एवरी सोमवार को each and every सोमवार हर सोमवार को एक मार्केट विलेज मार्केट and in the village market they bring cows buffaloes to sell. तो जो cow और the buffaloes which are very lean and thin कुछ दिन बाद मरने वाला बहुत एकदम दुबला पतला सा very lean and thin cows and buffaloes they make diaspora they make the media and local leaf paste. इस plant का जो leaf है उसका paste बनाते हैं they make a paste of that and they put it on the body on the skin of the cow and buffalo. The cow and buffalo they get allergic reaction, inflammation होता है और एक तो भागता करता है and they take it to the market and sell it at 80,000 rupees. So the buyer who is buying the cow and buffalo he thinks it is a very healthy cow or buffalo और घर पे लेके आ जाते हैं. After 3 days when the inflammation subsides they find out इसका कीमत तो 8,000 भी नहीं और हमको 80,000 इसको खरीद के लेके. So they are making money from immunology and you are reading immunology or reading QB, right and other books, but see, without knowing single hypersensitive reaction, they are making money, not the village people. So that is the beauty of India. So each and every location, as far as interiors, you find something new, discovery over there. So then I started doing more research about this, and even this plant was used as not only altitude indicator but mineral indicator. जहाँ पे gold होगा, जहाँ पे silver होगा, this plant start growing over there. There are types over there. So when we took this flower of this plant, this is the flower of this plant, but we have to use N95 mask because the flower को जब हम boil करेंगे, because if you boil the flower over here, you start getting irritation in your gums. तुरंत gums में irritation शुरू हो जाता है because that is not point. So we use the mask and when we boil this flower, but when we selected this flower in Ethiopia, in Africa, this flower is used as anti-rabies. If there is rabies infection, it is used for anti-rabies infection over there. And this is very, very important flower. In different places, you can see over here. Dog bite. Huh? So this plant is used as a dog bite. When there were no vaccines, this plant was used as a rabies. Next. So when we took this flower extract of this plant and leaf extract of this plant, we wanted to see whether it is able to synthesize gold nanoparticle or not. And to our surprise, we found that this particle, this plant is not only synthesizing gold nanoparticle, but it is synthesizing gold nanoparticle of a very unique geometry. See the geometry of the nanoparticle. This is a hexagon, and each and every face of hexagon is of the same length. Just flower from a plant, and it knows how to synthesize a hexagon of same length. See the triangle, equilateral triangle. Each and every side of the triangle is of same length. Equilateral triangle. Okay, so it does not have a ruler or scale to measure, but it is synthesizing of equilateral triangle. Next. So we wanted to see what it is synthesizing. In physics, if you are from physics, you might be knowing there is a concept called Oswald writing. So we have a couple of slides. Within few minutes, we'll finish. And you are hungry. Okay, so in case of physics, we have a concept called Oswald brightening process. So what is Oswald brightening process? Very small nuclei particles, very small particles are initially formed. They come together to form bigger particles and then their shape and size is changed. But this is theoretical process. It is very difficult to see in practice. In reality, it is next to impossible. Theoretically, we can very small particles are coming, they are coming together to form bigger. But this media blocker showed us exactly Oswald brightening process. We have very, very small particles, they are coming to form bigger particles over here. And finally, these bigger spheres are coming together to form triangles. So these smaller particles are around 12 nanometers in size. And when they are coming to form spheres, the spheres are around 60 nanometers, 59 to 60 nanometers. And when they are coming to form triangle, then they are around 286 nanometers. So this was a very interesting process. So now we thought, Biologically, when green synthesis, this is the only demerit or disadvantage. We cannot control the size and shape. That is the only disadvantage of biological. So we thought, can you control at this stage if there is a hierarchy, छोटा से बड़ा हो रहा है, size is changing, shape is changing. Can you control over here? Next slide. So here we started control. You can see when we added the good chloride with the media blocker leaf extract, GGA media blocker leaf extract. The nuclei particles are formed, very small, the nucleation was there. Now when there was a particle growth, when there was a particle growth over here, at this stage, when there are particle growth over the smaller particles were there, they started growing, we changed the halide ions. When we added chloride ions, we got triangle. As soon as we add bromide ion, we got nanopentagons. When we added iodide ions, we got nanowires. When we added fluoride ions, we got nanospheres. So this is the way we busted the myth that the green synthesis has no control over the size and shape. We could actually control the size and shape by using halide ions. By changing the halide ions at this stage, we could do this. Next one. So now you know different types of drugs are there in market, different types of cancer drugs are there. But if you see this particular component, curcumin, every day we are using turmeric, haldi in our food. Each and every food we generally use. But you can see, if you see curcumin, a single molecule of curcumin, it can prevent so many different types of cancers. A single curcumin molecule, it can prevent so many different types of cancers. Now you can see today the cancer cases are increasing like anything. Every family, some family member or relative, you will know that they will have cancer. Why? Because slowly we are shifting towards a diet where there is no intake of curcumin. Now, if you go to the noodle, if you go to Manchurian, if you go to burger, there is no curcumin. There is no it means application of haldi or turmeric is understood. So previously, if you go to old people, they can know हम लोग कितना कुछ करते थे, वो काम में कैंसर में होता था because whatever addiction they had at the same time they used to have food that was highly loaded with turmeric. Now, if they are moving towards the Western food or Chinese food, they do not have any curcumin content. What is the demerit? What is the demerit of this curcumin? This curcumin is non-polar in nature. पानी में नहीं थी, curcumin is never soluble in water. It is highly non-polar. So how can we make available this curcumin? Curcumin gets solubilized only in oil, only in ghee, only in butter. So if you see the cooking process of our grandma's time, even our grandmothers or mothers, how they cook, they will always take oil or ghee in the pan and then they add this haldi or this tarka and then why? Because there is sequential extraction of the curcumin. And after that, they add water. So that causes micro emulsion over there. So much physics and chemistry is happening in the product. We never think of that. You can never see that somebody or your mother or grandmother first add water and water addition के बाद एक चम्मच उसमें हल्दी डाल दी. You will never see. Always turmeric used to be added in with oil, ghee, butter like that, and then water was added at the last. Because first they used to extract the curcumin, that is the sequential extraction. Okay, the fractional extraction you are doing. And then you make a micro emulsion after adding water. Now it is binding. So this curcumin, one single molecule, how it is important? So if you take 5 grams of curcumin, 5 grams of curcumin in milk, only in milk, not water. Any kind of cancer, if you have any kind of cancer, that will stop at that stage. It will not allow the cancer to progress. It will stop. It will halt it. But on condition 5 grams in milk, because milk has fat. So milk has emulsifying property. Then only it can extract the curcumin. And why curcumin? A single molecule of curcumin, you can imagine it can control so many genes in our body. So all those brown genes are cancer-causing genes, pro-cancerous genes, oncogene. So those genes are down-regulated by a single molecule of curcumin. And those blue genes are anti-cancer genes. So those are upregulated by curcumin. So that is the power of curcumin. The next slide. Yeah. So what we did, we conjugated curcumin onto iron oxide nanoparticle, and those particles were around 15 to 20 nanometers. What we saw over here, you can see 90% inhibition of the tumor. When this curcumin, we try to make it available using iron oxide. Now, why we are using iron oxide? See, iron oxide is magnetic. If you inject the iron oxide over here, and I have cancer over here, I can put a magnet over here, all drug molecule will come in this side, it will not circulate throughout the body. That's why we will be a magnetic nanoparticle for anti-cancer therapy. You can say 90% inhibition of the tumor, 90%. And it can scavenge these free radicals. You can see 100% scavenging of super. 100% scavenging. So the free radicals are responsible for cancer. So next. So in conclusion, I think because of the incompatibility it has come down. So in conclusion, you can see now the biogenic nanoparticles, they are more biocompatible, they are safe, and you can use them not only for the anti-cancer, antimicrobial, but also for prevention, not only for the control, but also prevention of this. Also, you can use for medical imaging, also biomedical imaging, diagnosis, and treatment. Next. So you can see the COVID, during COVID-19, the recent pandemic, COVID-19, coronavirus is not the first time that it has come. Coronavirus has existed from long ago. So if you see the first stage of coronavirus, that was SARS-CoV in 2002, 2003. There were 800 deaths. Throughout the world, there were 800 deaths. And how many were infected? 8,000 were infected. 8,000 were infected and they were 800. So 10% mortality was there at that time. But see now, March 12, when this Middle East respiratory syndrome was there, March 12 in 2012, after 10 years, 10 साल के बाद जब नया कोरोना वायरस है, March 12, you can see the date were 858. Now you will say, उतना तो बढ़ा नहीं, it was 800, it became 858. But see the number of infections. It has become more dangerous, it has become more virulent. Okay, the infected were 2,494 and they were 850. And see in 2019, okay, so after 7 years, when the new one came, another one came, the SARS-CoV, you can see the date, imagine the date, 70 lakhs for the day. So, probably after 7 years or 10 years, it can again mutate and a new COVID can, a new coronavirus can come. Now, why there was a major failure? Because if you see this coronavirus, the size is 120 nanometers. If you want to stop it, we have to design some drug which can match the size of the virus and it can go and stop. So what Israel and Sonovia did over here, they created zinc oxide nanoparticle and they coated the fabric. So the coronavirus wanted to penetrate to that because of the size it is matching and it got stuck over there and it was killing the coronavirus. So now the nanomedicines are smaller nanomedicines and they are used as per the size of the virus. It is customized numbers. Next. So with this, I just so these are the books that I have edited. So if you want to go to the, if you want to start synthesizing your nanoparticles, if you want to understand nanobiotechnology or dive into the arena of the nanoparticle researches, you can go through these books which are related to nanobiotechnology for agriculture, nanobiotechnology for medicine, and different all different areas. Now we are editing one book, it is almost, it will come in another 2 or 3 months. There is nanobiotechnology for environment. Next. So with this, I thank the organizers of this conference, Principal Sir, Mr. Dharmani, and then organizing secretary, Dr. Sougata, and all dignitaries, faculties of the college, and all participants. These are students who helped us in the research that I showed, and these are the funding agencies with the help of this funding we have completed this research. Thank you so much, and I will be happy to join you.
Thank you so much, sir, for your enlightening session. May I now request chairperson of today's session, Dr. Ritesh Chandra, to conclude. Oh, sorry, any student, if you have any questions?
Hello. Good afternoon, sir. Sir, as we all are adapting the foreign culture, foreign food, and as you mentioned that the Chinese and Western foods are not providing continuity. And that is the reason in this generation why cancer is increasing. So if we are performing the same, we are eating same food, we are adapting the same lifestyle, then why there are less number of cancers in the Western culture or the Chinese culture, where they are also not utilizing perfume. Either if you go to US, also all hospitals are full of cancer patients. US has a huge number of cancers, huge number. They cannot control the cancer thing. And that's why California, they have started introducing something called turmeric latte. Good healthy that we are using over there. When it goes to US, it becomes turmeric latte over there, $20. Yeah. So we have chocolate latte till there. But now they have introduced turmeric latte. Whatever you get over here at 20 rupees, 50 or 30 rupees, you will buy over there at $20. So you can imagine the thing. So they have now started accepting the Indian culture. They have a lot of cancer cases, but at the same time, their medical facility is also getting upgraded. They are having customized immunotherapy. So that's why you can see even Rishi Kapoor was taken to US to have immunotherapy. But unfortunately, he could not survive over there. So this is the thing that are happening. So at the same time, even Indians are trying to get the same medical facility, but US is unable to give them the treatment. So cancer is present everywhere. Thank you.
Very good morning, sir. I have a question that you tested the nanoparticles on the NCS service already, the breast cancer service, and it got very positive results. So how would you interpret, like if I'm using some other satellites like Indian 31 and other things, then how should I interpret the results? Like what would be the variation or how should I testify the same?
Yes. So when you are using those cells, you are considering about triple negative cell line. So at the same time, when you are using those cell lines, you can use different markers like the gene expression, the tumor-specific antigens. So whether those antigens are getting expressed or they are getting inhibited. If you can see they are getting inhibited, that means it is a positive marker that it is unable to increase. So here we have to add two more experiments, that is either RT-PCR based experiments, quantitative PCR, or you can also add question blotting based experiments to see the gene expression. So I think that can be useful.
Thank you, sir.
Anyone else?
सर मैं नैनो टेक्नोलॉजी में अभी नया स्टूडेंट हूँ, बट आपने जो चालू किया और आपने जो अपनी स्पीच में बताया अबाउट द इग्नो मेडिसिन, तो बेसिकली आई एम बॉटनिस्ट और इग्नो मेडिसिन पे मेरा पूरा काम भी है, और यू आर जस्ट साइटेड एन एग्जांपल ऑफ कुकिंग. तो हम जो किचन में जो और मसाले यूज करते हैं लाइक जीरा, अजमा, so everybody is using this thing in regular form. तो सर उनसे भी थोड़ा प्रकाश डालिए. तो ये जो सारे बच्चे मेरे पीछे बैठे हैं, जो घर पे जाके बोले मम्मी आज आपने जीरा या कुरकुरे डाला है कि नहीं, सो इट विल गेट एन इम्पैक्ट. और वो भी अपने पेरेंट्स को समझा सके कि ये हमें आपको मत दो. और दिस आर द जनरेशन जो अपने बच्चों को और भी ज्यादा आगे नए सोफिस्टिकेटेड और एडवांस टाइम में बच्चों को नरिश करने वाले हैं. तो अगर इनको पता चलेगा तो आगे जाएगा। सो प्लीज सर आप जरा उनके बारे में बताइए मेरे बच्चों के लिए। सर ने बहुत एक इम्पोर्टेंट बात कही, तो दैट इज हाउ वी कैन गो फ्रॉम किचन like kitchen spices to ऐसा medicine को convert कर सकते हैं। There's a shop in US called Walmart. In Walmart, the whole section is based on spices. India में इतना spices है हमारे पास, but they have capsules for black pepper. They have capsules for cinnamon. They have capsules for cardamom. इलायची से शुरू करके सभी चीज का कैप्सूल है। They have capsule everything. Whenever every year when you go over there, I see turmeric capsules and turmeric pepper capsules, black pepper capsules. Ginger का अलग मिलता है, ginger और they have a completely separate section for ginger capsules. So definitely उनके खानों में they do not have those medicines spices. That's why we have to take them as extra supplement, additional supplement. When I go to their house, suppose there is a friend and they have table. जैसे हम लोग यहाँ पे कीनो डाइटिन वगैरह रखते हैं, they have those capsules. हाँ, दिन में एक सिनेमन कैप्सूल ले लिया, एक कार्डमम कैप्सूल ले लिया, पेपर के कैप्सूल ले लिया. So they have to take it from external. But you can imagine each and every of this flow oil having. Okay, we have cinnamon oil. So definitely these things can be used as bioemulsifying agent and that can be taken as food supplements. But हमें supplement के हिसाब से क्यों लेना है? We are already included in our dietary regime. Understood? So make sure that ऐसा नहीं है कि अगर लेना है तो some of you do not have any disease, but every day you are taking 5 grams of protein. Then the body will become resistant to that and you develop some complications. इससे जितना हमारे food dietary regime में है, that is okay. If a person is having cancer, then that person is to take 5 grams of protein. Unnecessarily, किसी को लगता है YouTube में देखा, root change करने के लिए बहुत ज़्यादा करके. Then definitely will develop something. Why our food regime has been created in that way? Because it is as per our हमारे body के जो composition है, as per the geographical location, as per our conditions of the body, it is developed. In humans, they do not have those things. That's why they are doing. So whatever you have, you can take it as a balanced diet, and then there is no problem. And there is a huge scope of developing each and every. जैसे हमने curcumin को functionalize करके nanomedicine बना है. You can develop your nanomedicine. Take one nanoparticle, zinc oxide. You conjugate the chemical from cinnamon, you conjugate the chemical from clove, you conjugate the chemical from any kind of your masala, and then you can definitely get a new novel medicine with better chemical. आपकी के लिए अच्छा रहेगा, and your dissertation also. And I will just show you some source. Similarly, we are doing with the green tea. You know a component called catechin. Green tea, there is a component called catechin. And just now we conjugated it on titanium dioxide nanoparticle and we published in a very high impact factor paper because that is more available and more antioxidant and we are conjugating on titanium dioxide. So you can think of such kind of research and it would be really very helpful.
Anyone else?
So, I just want to ask you, it's an AI technology and computer aided design that we doesn't have to be on that, like we have to mix up the two kind of article and what we structure will be form. So, is there any AI technology, computer aided design forming available very right now that we just to the computer that what is the new form maybe big like that?
Yes. So now a new technology has come that is AI driven QSAR of the nanoparticles. So you already there is a lot of data that are reported. If this is this shape, if this is this size, we have this much activity. So huge number of data is already deposited. Now people are not doing any laboratory experiment. They are retrieving the data from there. Based on the AI, they are developing quantitative structure activity relations of the nanoparticle. Previously, QSAR was done for the chemical compound. So chemical structure है, benzene is है, कुछ दूसरा compound है, is to do this one. But now, since there is already a library available, so they are already taking this. Now, if you see, I don't know whether you have seen through the ICMR web page. Kali last date AI driven solution for healthcare. Kali last date ICMR project submission. 13 February. So now people are giving TCAS and different other government even AI, they are asking for new proposals that are AI driven. AI driven drug development of drug discovery. So definitely, if you do not have lab facility, you can already retrieve the submitted data and you can have a very good research on this. That's why that is the main aim of our table. So since we have already developed a table, now people can use that table for AI generated assisted drug development or composition analysis.
Thank you. Anyone else?
Sir, so we nanoparticles and it's mostly are made with any metal like iron, copper, zinc, or titanium. So nanoparticles and nanomedicine are used for in this application, mainly in vitro conditions. It is in future it will possible for any treatment in any kind of disease like cancer. So we एक्सरे का प्लान था जैसे जो जैसे कोको में तो उसके साथ मेटल्स भी जाएगा बोलूँ तो नहीं जाएगा
I was expecting.
तो
Maybe
side effects from
I was expecting the other question to me. So this is very important question. What about the toxicity? This is very important question. That if अंदर जाएगा फिर इसका effect क्या है? Now if you see for gold and silver, I told anti-biofilm activity. Cancer के लिए I have not told you about the gold and silver. Cancer के लिए I told you iron oxide. Now there is a reason for this. If you go to the market, अभी भी आप suppose you go to Aditya any clinic, medical store, and if you ask silver sulfadizing है क्या, you will get the silver sulfadizing cream. आपको मिल जाए. Why silver sulfadizing is used? Silver sulfadizing is used for the burn wound treatment. It is used as a topical application. So that's why if you see my application, I told anti-biofilm application. Biofilm कहाँ पे होगा? अगर हमें burn होगा, हमारे बाहर होगा, in the outside, in the external. So for external application, we can use gold and silver because silver is already in medical use. So there is no question of toxicity if we are using outside. So there is no problem. Okay, silver you can use. Silver sulfadizing is already available in the market. Now, if you take inside, which kind of nanoparticles we are talking about? Iron oxide. Hemoglobin में क्या है? Iron is there. So there is no question of toxicity over there because iron is already present inside our body. So if you are taking this nanoparticle, it will metabolize and it can be used for our iron intake and iron metabolism. Second thing, if you are seeing the linker मैंने क्या use किया? Citric acid. Citric acid is an integral part of Krebs cycle of TCAS cycle, tricarboxylic acid cycle. So there is no question of toxicity because of citric acid addition. Third concept drug मैंने use किया, it is dioxin. You get huge amount of dioxin in methi, phenolphthalein. Dioxin is a part of our regular intake of our food. So nothing is there which can be toxic. This is one thing. But when you are taking intake, when you are considering drug intake, then you have to design rationally. मैं gold या silver को मैं conjugate करके कुछ नहीं आऊँगा. Now, gold या silver या दूसरा कोई metal डालो तो क्या होगा? You have to take care that metal nanoparticle is below 80 nanometer. If it is below 80 nanometer, it can pass the glomerular filtration of the kidney. On condition 80 nanometer to नीचे होना चाहिए. If you see our gold, silver, bimetallic nanoparticle, I told you it is 15 nanometer. So it you are taking those kind of nanoparticles, which will work in your body, and within 24 hours or 48 hours, through the urine, it will be passed out. There is no question of accumulation. So these are the three possibilities how we can get rid of the toxicity.
Thank you, sir. Anyone else?
Uh,
Sir, as you mentioned birthmark, sir, do you think that the visual birthmarks are a form of nanomedicine?
The thing is, nanomedicine is something that is like nano dimension, 1 to 100 nanometer. So definitely when you are going for traditional plasmas, they are very minute particles. So they are always in a form of nanomedicine, but they are not all, in case of nanomedicine, we very well know the component or composition of the nanoparticle, which nanoparticle is. But in plasmas, they are complex nanomixtures. So we do not know in case of plasmas, we do not have a single kind of component in it. We have a lot of things inside that. So although they are in nano dimension, we cannot call them nanomedicine because for medicine, for medicinal purposes, we should know the composition. So that's why our Ayurvedic medicines or homeopathic medicines are not available in the Western world. Because in Western world, when you are selling a medicine, when it is available in medical store, you have to write the entire composition of the medicine, what is present, how much milligram, how much milligram, what it is. So in Ayurvedic medication, they are already in a nano dimension, but they cannot be coined as the term nano medicine because we do not know each and every component of those medicines.
Thank you so much, sir.
सर आपने सिंगल गोल्ड और सिल्वर न्यूरो पार्टिकल का बनाया है और जब आपने बात की कि वो कंबाइन जब हम करते हैं सिल्वर और गोल्ड तो ऐसा कोई आपका स्पेसिफिक ऑब्जर्वेशन है कि रिजल्ट जो मिला उसमें कंबाइन वाला क्या करेगा या सिंगल वाला कम मिलेगा सर? हाँ, जब हमने कंपेयर किया तो हमने देखा कि जो गोल्ड और सिल्वर का जो कॉम्बिनेशन है गोल्ड और सिल्वर से नैनो पार्टिकल क्योंकि उसका 50 नैनोमीटर साइज है उसका जो एंटीबायोटिक इफेक्ट है उसके साथ इसको मैंने नहीं लिखा है हमने उसका कंट्रोल ओनली गोल्ड एंड ओनली सिल्वर करके ऑलरेडी हमने एंटीबायोटिक किया सो ओनली गोल्ड का कुछ भी एक्टिविटी नहीं था गोल्ड इज वेरी बायोकॉम्पैटिबल तो उसकी एक्टिविटी बिल्कुल भी नहीं सिल्वर का था बट सिल्वर एक्टिविटी वाज 10 टाइम्स लेसर देन कॉम्बिनेशन तो इसीलिए जब कंबाइन हो रहा था गोल्ड वाज हेल्पिंग इट टू गो इनसाइड और सिल्वर वाज हेल्पिंग इट टू एनहांस द एक्टिविटी तो दोनों का मिला के इसका जो सिनर्जी था बहुत अच्छा था
Thank you so much, sir. I suppose that's why silver is being inflated nowadays. Sir, my question is about diaspora bulgy failure. Is it, are you talking about swan basma or like that, in that it is made up of that?
Diaspora is something sort of funny. So you can prepare sort of from diaspora. तो उसमें डायस्पोरिया नाम डाबर चंपस के अगर बॉटल में देखेंगे उसमें लिखा रहता है वारा ओनली दैट इज वारा इट्स नॉट मेंशन डायस्पोरिया ओनली है but it is वारा so from that sort of basma is prepared from that other basma and different version of basma so these three basmas are very common from diaspora but here in this whatever I do these are not in basma form these are pure nano but हमें आइडिया कहाँ से मिला कि ये स्वर्ण भस्म के लिए यूज होता है सो प्रोबेबली इट कैन प्रोड्यूस गोल्ड नैनो पार्टिकल्स सिल्वर नैनो पार्टिकल्स इसको आइडिया मिला तो इंक्रीज कर
Thank you so much. It was very wonderful, enriching, very strong session. May I now call upon Dr. Ghosh, chairperson of today's session, to participate, sir. Thank you so much, sir. Really wonderful session. And I suppose students are not interested in nanotechnology. Thank you so much. Now we will proceed for lunch and after that we will have validation session.